Thank CADD organized this nice symposium for sharing my teaching experience using the Schrodinger package for up-level courses. Here's the outline of my talk. The first part is general introduction on the computer-aided design. And my teaching philosophy is first to motivate the student on the thing we're going to teach. If students are motivated, it's much easier for them to learn. The second part of my talk is my rubric and syllabus to cover the content in the computer-aided design. Okay, so let me first convince you that computer-aided design is a desirable courses for the future of our students. Why computer-aided design matters? Traditional way to develop a drug will take $1 billion, 15 years for developing new drug because most of the time resources spending on the bad compound. If we can remove the bad compound before we do the wet lab testing, and then we can greatly cut the cost and time for developing the drug. That's the purpose of the computer-aided design. We fully take advantage of the information of our drug target. For example, their sequence, their structure, their activity, and then to filter out the bad compound and save a short list of compounds for testing. So here is a workflow we implement using the Schrodinger package. We'll start the structure of drug target, and then the library with 70 million compound. So we first use drug property, filtering out some of the bad compound with bad drug property, use the Schrodinger quick prop. And then we do the glide docking to filter out the compound has a bad docking score. And the remaining compound will do the molecular scaffold clustering, use the canvas. And we pick the final 20 compound, and which not shown here. We will further do molecular dynamic simulation and the post-simulation analysis and identify the top compound. Okay, with the top compound, we'll pass to the wet lab for testing protein-based activity, cell-based activity, and then animal-based activity. So the first part I typically cover is the database search. And what's the basic database? It's a big spreadsheet with a row recording the interesting entity. And in this case, it's just the student information. And then the column is also called attribute, defines the different aspects of the entity we're interested in. And then use the keywords, which are the attribute plus Boolean operator. We can search subset of the record we're interested in. Okay, so with this understanding, I will introduce students to do the protein sequence search, use uniprotein database. To search the protein family, use the interprotein database. And to search the protein structure, use the PDB-SUM database. And then to search the literature, use the Web of Science. In last year, we also teach students to use ChatGPT. Now, ChatGPT really can offer the very professional answer to any type of question in any domain, almost reach the professional level. Yet, ChatGPT not able to generate interesting question. And so that's the part I train my students to ask the challenging question. For example, here, how to insert a artificial gene into human genome use a CRISPR-Cas9 tool. And ChatGPT will provide a procedure and ask you students to find more information and to convert that into the diagram like here, okay, which shows the student understanding this process. So for that, we need a plasmid of the CRISPR-Cas9 and also has coding the guiding information, which will tell the place to insert the gene into the human genome. And then we need the recombinant gene we want to insert. Okay, so with the plasmid with the gene, it can insert into the target cell, and then this plasmid will translate into the protein with guide sequence, which allows to bind correct place of the human genome, and then the Cas9 will cleavage to create the break, and then the insertion gene has the matching sequence to bind the broken site, and then the repairing from the whole cell on this break point, and that leading to insertion the foreign gene in the human genome. So with this kind of thought experiment, and student will demonstrate the conceptual understanding of the subject of the question they try to solving. Okay, so homology modeling is a basic task in modeling the protein structure when there's no structure for the target sequence. And for that, and we can find the homolog, which the sequence are similar to our target sequence and whose protein structure are available. So then we can use that template structure to build the structure for our target sequence. Molecular docking is a powerful tool given a drug target structure, given the active site, then we can use molecular docking program to dock the ligand into the binding pocket and output the docking score. And if we're repeating this for a large library of compound, then we can sorting based on the docking score to pick top hit. And that's literally high throughput virtual screening. Molecular dynamics simulation is another powerful tool. And here, and we see the transformer change shape. And we know the underlying physics is the interaction between the different parts of the car, and which are described by the Newton equation. So if we want to model the dynamics of the bimolecule, we can develop the parameter defines the molecular interaction, for example, bonded interaction, long-range interaction like electrostatic, van der Waals interaction. With all those parameters describe those interactions, we can apply Newton's law to given initial position and find out the force. And from force, we can calculate acceleration. From acceleration, we can calculate velocity. From velocity and initial position, we can calculate out future position. So if we're repeating those procedures, we will get motion of the molecule. So in the movie, it's just to show you folding a small helical peptide. I want to give you a brief history about molecular dynamics simulation. It literally started 40 years ago by Dr. McCammon, used the Apple computer to simulate some particle in gas phase for a very short period of time. In 1998, my PhD advisor, Dr. Duan, was able to use a supercomputer to simulate a small protein in the water box for almost a million-fold longer time. And you see, it's not fold into the native structure yet. This work inspiring Dr. Xiang, who invest money to develop specialized supercomputer to do the molecular dynamic simulation. And now you see it can fold small protein into the native conformation. Even without access to supercomputer, now with the graphic card, we can do decent computation. Another advantage is that if we do nothing, our computing power will keep increasing every year and the price will drop every year. So here, when I was postdoc, and I tried to build a small protein which has both the helical second structure and the beta sheet, beta helping. And you see the helices fold first. And then the helpings start to forming and assembling a lot of the wrong conformation before it finally found its correct conformation, which is supposed to be the lowest free energy state, and then will stay longer time there. I also simulated the interaction between the drug with drug target. In this case, this is with the first line chemotherapy drug, and it's work by insert into the double-strand DNA and log the genetic information. From simulation, we kind of can see how the drug insert into the drug target. Okay, it's a lot of the induced fate kind of bonding, just interaction. So now base pairing form. We also can use computation to understanding how the engineering T-cell receptor work and how to improve it for the treatment. So let me first give you some background about this chimeric antigen receptor T-cell therapy. What it does is it takes the blood from the patient and isolates the T-cell, and then engineering the T-cell receptor into the T-cell and culture the T-cell with the specific CAR for large population and insert back to the patient. And those T-cells were killing the tumor, leading to the cure. Here's an example. The Emily basic diagnosis 10 years ago with leukemia, which basically will claim her life. Fortunately, he took the T-cell therapy, and here's how she is. She's the T-cell treatment demonstrates the first case of the cure of the cancer, and also potential lifelong immunity. And here's the 6 T-cell treatment approved by FDA to treat blood cancer. And if this benefit can be extended to the solid tumor, that will bring the benefit to much bigger population. Yet, no success has been achieved on the solid tumor due to lack of the selectivity and sensitivity. Well, how to improve that? We first have to try to understanding how it works. The CAR is recognize the antigen on the surface of the T cell, and then that bonding will activate the signal domain, which are here shown in the green part. And the lily, it has immunotyrosine active motif, which basically has a 6 tyrosine radio. When it's phosphorylated, then the receptor is activated and the signal is transduced from the outside cell to the inside cell. How that work? And this is the
most popular model to suggesting the activation. And this uses native T cell receptor. Okay, so it's a more complicated CADD, but the activation should be very similar. So the CD3 zeta, which are the signal domain, which basically will bind to the membrane through the cell bridge with the basic residue in the protein and then acidic phospholipid. And upon the bonding with antigen, it's an induced conformational change and to leave the membrane to expose to solvent. And then kinase will come here to do the phosphorylation. So this is a very sound kind of model. Yet, involvement mutation data does not support this model. Based on this model, if we mutate those basic residue to make the peptide break the cell bridge, it will be exposed to the solvent, and usually it will increase the base rate phosphorylation. And yet, the experiment suggests otherwise. So that's the motivation we want use the molecular modeling simulation and try to understanding the activation mechanism.
And the CAR we pick is a CAR which are targeting on the HER receptor, which are biomarker on many solid tumor. And this anti-HER2 CAR basically show developed to treat the HER2 positive breast cancer, and it has been shown promising the cytotoxic to human HER2 positive breast cancer in mice model. And here's is a component. It's literally made from the cellular protein. However, the high resolution structure are not available except the antibody part. And the purpose of this study is try to build a homogeneous model of this receptor and probe its structure dynamic to understanding its signal transduction mechanism. So here's what we did. And we built the homogeneous model, and then we built this receptor, and this is the antigen, and grab from the crystal structure. So with that, then we can do molecular dynamic simulation, and actually from the molecular dynamic simulation, and we can easily see how the signal is transduced. So let's take a look at this, the APO system without antigen. And you kind of can see that this is a extracellular antibody domain is very flexible to search the antigen, and this portion is much more fixed and less flexible. And yet the story change once it's bound to the antigen. So now you clearly see this portion are fixed, and then this intracellular part are moving large range and search for its protein partner. And we're believing this is the signal transduction mechanism. So in other more obvious way is that here you can see, and we plot the center of the axis antibody domain, and green are the transmembrane, and yellow dot are center of the signal domain inside. So this is the APO system. You can see the extracellular domain are flexible, search large space. So when it's bind with the antigen, and then it's become fixed, but the signal domain now search large space, become more exposed solvent. Besides that, we also observe conformational change. So this is the second structure plot, and we kind of can see that in the APO system compared with holo system, lose the helical structure. Okay, which actually help it to convert into the conformation, make the kinase easy to do phosphorylation. So this is the actual structure of the kinase with its substrate with tyrosine here. And our analysis show here, yes, indeed, the 6 tyrosine and their conformation change to more active conformation. So that's the intraconformational change upon the antigen bonding. So here's the mechanism we propose. It's called the bonding-induced domain flexibility switch. So before the bonding, this is the HER2 and the antigen, so it's very flexible and assembling the large space. So for the antibody, but once they bond, they become more fixed position. And then the intracellular domain now it's a search its partner moving large space. And once they make it easy for the kinase to bond and do phosphorylation. Okay, this mechanism actually just mimic what we learned in high school called the copper pendulum. And you see the energy can be transferred through this connected soft strand. So, okay, this energy stop and energy transfer inside. Okay, so the supporting strand really is the transmembrane and then flexible part. Other protein work the similar mechanism? And answer is yes. Here we found the membrane receptor kinase, and they mimic the same architecture, except they have some repeat in the extracellular domain and also intracellular domain. And we hypothesize this will improve the sensitivity, selectivity of those signal transducer receptor. If that's true, and then we can borrow the idea to optimize the car to have the repeat in extracellular domain and intracellular domain. And this actually has been successful implemented by a UCLA group to develop bi-specific 10 CAR. And in my group, now we try to develop this tetra-specific CAR and targeting the solid tumor. And are there other evidence to supporting our signal transduction mechanism? So if follow the CARP pendulum, the transmembrane and flexible part, which are basically serve as the flexible strands supporting the transfer the energy, so which should not have the conservation, so much conservation on that sequence. And that's what we found. So if you cut those portions, and you can see, and their sequence much more diversified, except the transmembrane portion, and then lengths are very different. So this provides another piece of evidence to supporting our model. This is another application of the virtual screening, which use the workflow I introduced and to find the short list of the potential compound for well lab testing. So this protein target is for the TRIP-negative breast cancer, which is the hardest form trait for the breast cancer. Okay, so through virtual screening, we identified the 6 compounds. Then in my lab, we did the well lab testing, the enzyme inhibition assay, and we found a compound which give us the inhibition IC50 10 micromolar, so very decent hit. And usually it will take the screening 10,000 compounds to find one, but now we only use the 6 compounds, we found one. And then we do the cell viability testing. It shows the EC50 effective concentration at 50% killing the tumor cell. It's 10 micromolar. And this compound is ready for the animal model testing. Okay, So now we come to how I teach the computer-aided drug design in the up-level courses. So here's my rubric for my courses. And every student will assign a research project, and students will do the 2 oral presentation and plus a written report. And 1 oral presentation basically will publish in the social media. And then student will use the Schrodinger exercise, learn the basic molecular modeling simulation technique. And then they will use those techniques to do their project. So here's the detailed schedule I have. So the database search, and then metro to do the protein preparation, and to do homology modeling, to do the docking, and to do the virtual screening, and to do molecular dynamic simulation, and identify the drug candidate for their drug target. Okay, so here's drug target. 1 year we implement the class A GPCR, which are largest class of the drug target for current medicine. No surprise, 2 years ago we picked the COVID-19, we picked the SARS-CoV-2 target, developed the anti-SARS-CoV-2 drug. And this year we picked the kinase as the cancer target. And here's the YouTube video student post their research talk. And in the past 8 years, our students present over 100 posters, and students publish 40 papers, over 40 papers. And we also had students learning those things and doing remotely. So Lauren basically our UCSB student, which found CADD interesting. So we through the remote teaching, and he learned the technique, published 2 papers. And here are students from Indonesia, and learned the technique, and we published several papers. Here's the acknowledge for over the past 80 years, over the 300 students have to use Schrodinger to learn the molecular modeling and simulation for computer-aided drug design. And with their effort, I can present the story here. Besides that, I'd like to thank you. The Schrodinger Education organizes next workshop. And I also like to acknowledge our funding agency, in particular, the Antigen 2 supercomputer allow us to do the simulation for the very large system. And here's a picture of some of our students. Thank you very much.